Thank you very much for the opportunity to talk about our work with HIV and SARS-CoV-2 here with our group at Rockefeller. So as all of you know, the idea of transferring antibodies to either prevent or treat infections has been around for a very long time. And I'm illustrating this here, showing data from the time when anti-diphtheria serum was used prior to the availability of an anti-diphtheria vaccine. And as you can see, over time, the incidence rates really decreased quite dramatically. In addition to diphtheria, many other infectious diseases have been prevented or treated by the antibody transfer before each of these vaccines became available. And it was this in mind that HIV antibodies and SARS-CoV-2 antibodies were pursued. Antibodies are interesting molecules that have different functions. Antibodies can neutralize virus that are free in circulation, but they can also interact with the immune system to trigger either killing of infected cells or enhancement of other arms of the immune system, and altogether work to clear an infection. Antibodies are induced following HIV infection, and including antibodies that are neutralizing and are broad. And this happens despite the very intricate nature of the HIV envelope, particularly despite the high diversity of HIV envelope. What happens is in the subset of people that live with HIV, over time, following this interplay between viral evolution and antibody response evolution, they go on to develop antibodies that are broad and very potent. But how can one identify out of the multitude of antibodies that are elicited against HIV, which ones have these optimal characteristics? So methods that were developed in Michelle Nutzensweig's lab and adapted by many other labs allowed us to have a greater understanding of HIV antibody responses. And that is done by identifying sorting out B cells that are binding, that are specific to different areas of HIV envelope, and going on to clone and produce the antibodies that each of these B cells can then produce, such that one can have a library of antibodies that can be carefully characterized to select the most interesting, potent candidates that could be tested in clinical trials. And by work of our group and many other groups, we now have a multitude of different HIV antibodies that have entered clinical development. And they're illustrated here where the envelope trimer is in gray, and the different areas of the trimer that are targeted by different classes of antibodies are illustrated in the different colors. Over the next the last 5 to 7 years, many clinical trials have been conducted by different groups, and I'm trying to summarize in this slide where we are in terms of our clinical experience. Earlier this year, we learned the results from the AMP studies, and we learned that an antibody can indeed protect from infection as long as the viruses are highly sensitive to that particular antibody. And then there are also emerging evidence from different clinical trials showing that combinations of antibodies can maintain virus suppression in an HIV-infected person in the absence of ART. Antibodies have potential advantages in contrast to standard ARV regimens because they are a safe class and they do not have the risk of selecting for ARV resistance. However, there are significant challenges in the implementation of antibodies. One of them, an important one, is the idea, the fact that there is a level of pre-existing resistance to antibodies, either within an individual or in circulation in the community. And also the fact that antibodies are still costly to make. When we turn to the potential effects of antibodies in HIV remission or long-term control, that is still pretty much an aspirational goal, however, and that will likely require combinations of antibodies with other immune-mediated strategies. However, there have been recently promising results from non-human primate studies that have given the support and then the background to launch different clinical trials testing different strategies. So in the next few years, we will be learning more about the effects of antibodies in HIV remission. However, a year and a half ago, we all were struck by the threat of SARS-CoV-2, and many of us repurposed our efforts to address SARS-CoV-2. And the experience with HIV antibodies really supported and accelerated the development, I believe, of SARS-CoV-2 antibodies, where techniques that were in place for cloning antibodies and characterizing antibodies to HIV were quickly adapted to SARS-CoV-2. And our group and many other groups have now have a greater understanding of antibody responses against SARS-CoV-2. First, we know that antibodies that are elicited against natural infection or vaccination, they typically target similar areas within the spike protein. And there are common types of neutralizing responses among convalescent individuals and vaccinees. Interestingly, and in contrast with HIV, in SARS-CoV-2, highly potent neutralizing antibodies can be elicited early after infection, and these antibodies have few somatic mutations, which demonstrates that it doesn't require too many passages through germinal centers for these antibodies to acquire activity. And this has to do probably in part to the structure of the spike protein, where the epitopes are more available to the immune system to generate such immune responses, and in contrast with HIV. Work by Pamela Bjorkman identified different areas of the spike protein that are bound by different classes of antibodies. And unfortunately, the picture on the left is a cut, but it illustrates these four main groups of antibodies. Antibody responses following natural infection, as I mentioned, and following vaccination targets similar epitopes on the spike protein. Also illustrate that that the areas that are target to neutralization also coincide with areas where mutations have been developed by the new circulating variants, showing that these variants are really emerging following immune pressure, either from natural infection or from more broadly vaccination. In parallel to better understanding of host immune responses, several clinical trials moved forward very quickly, such that here in the United States, we have 3 different options of combination of single monoclonal antibodies that can be used for the treatment of SARS-CoV-2. We've also learned that timing of treatment is critical for the effects of these antibodies. Antibodies have shown activity even in the context of post-exposure prophylaxis, and many other antibodies are in development, including antibodies that have longer half-lives. However, it's already been clear that emerging variants can impact the clinical efficacy of some of these antibodies that target those areas to which the variants have mutated. So if we take a step back and we again contrast HIV to SARS-CoV-2, while it took 10 years to the development of these newer generation HIV monoclonal antibodies to the results of the first efficacy study of antibodies to prevent infection, in just about 10 months, well, now a little over a year, we've had 3 different options of monoclonal antibodies to treat SARS-CoV-2 and many other antibodies that are in phase 2-3 studies, which again brings me back to this slide to illustrate how the 2 fields have helped each other, where the background technology in HIV helped accelerate the development of antibodies against SARS-CoV-2, and now the quick implementation and the testing of many SARS-CoV-2 antibodies brings back to HIV, because now we know that monoclonal antibodies can be very safe in diverse populations. We have data showing either both effects in prevention and therapy, and also it demonstrates the possibility of fast development and fast deployment of such strategies. With that, I would like to thank the group here at Rockefeller with Michelle Nussenswag, Charlie Rice, and Paul Bienage, that we worked very closely together on our SARS-CoV-2 antibody studies, and the many other collaborators across different institutions with whom we have been working over the several many years on HIV and our funders. Thank you very much.